and as of now, it's recording what's happening. And so I just would recommend that to extension educators because I think it's a really useful thing. So Dr. Gibb and I have decided we're going to break up this session a little bit, in part because I know not all of you probably spend your entire life thinking about beef cattle genomics. In fact, this may be the first time you've ever thought about beef cattle genomics. So I want to give you a little DNA 101 before we get into the nuts and bolts of it, because otherwise we'll be having a hard time getting through it. So without further ado, then I'll give you the kind of the DNA 101 lecture here. So hello. Are we going to work here? What's going on? All right, there we're good. So animal breeders have been using selection on phenotypes for many years, and I think you'll all agree that we've made excellent use of that technology and have made tremendous progress in both our dog populations, but also our livestock populations. But where we really started to make tremendous genetic progress in terms of productivity was when we started to use measurements and objective evaluations of animals. And when that got incorporated into dairy cattle breeding programs in the 1950s, 1960s, we really saw the genetic trend for milk production take off. And similarly, we've had analogous gains in the poultry industry and the swine industry, and perhaps to a lesser extent the beef industry, in a part due to its the integrated nature of it. But now we've got the opportunity to incorporate DNA information in addition to objective phenotypic records and pedigree information to improve our estimates of an animal's genetic potential or genetic merit at a young age. And that's really what the focus of this technology is. And so before we kind of get into the genetics, I just want to make sure we've got a basic understanding of DNA as a concept. And there was a rather amusing survey that came out the other day that was done by Jason Lusk at Oklahoma State University, where he asked 1,000 respondents whether they supported or opposed government policies for mandatory labelling of foods produced using genetic engineering. That, of course, is a very controversial and hot topic. But he also threw in one here that asked if they wanted mandatory labels on food containing DNA. And 80% of people said yes, they would like that. And so that's kind of, as a biologist, a little bit of a scary proposition, that 80% of the population don't know that all food contains DNA, whether it's organic or genetically engineered or conventional. So I will just remind everybody that every cell of every organism has the entire genome of that organism in that cell. In the case of a cow, it's got the chromosomes that make up the cow, 3 billion base pairs here, and of course the central dogma of molecular biology is that DNA is made up of these nucleotides, which are translated into RNA, made into proteins, and proteins is what we're made up of. And of course we share a lot of genes with cattle, and I always am amused by people that don't want genes getting transferred from one organism to another, because we have a lot of genes that are associated with cattle. But it's really here is where we're talking when we're talking about marker testing, because these nucleotides, the A's and C's and T's and G's that make up the universal triplet code for encoding amino acids, are where we have variation in the genome and that we exploit in order to be able to track genetic variation in animals. And so these single nucleotide polymorphisms, also known as SNPs in common vernacular, are basically just genetic markers that are located in the DNA of organisms. And so what is a genetic marker? It's basically just a DNA sequence variation. So in this particular case, there's a TC SNP here that's associated with phenotypic genetic variation in a population. And shown here is a well-known naturally occurring genetic mutation that's associated with myostatin, which gives you the double muscled phenotype that's typical of the Belgian blue animal. This particular animal's gene called myostatin is mutated, so that gene no longer, as the Latin name would suggest, myo, muscle, statin, stop, it no longer stops muscle proliferation, and so you get this tremendous proliferation here. Now, I don't need to genetically test that animal to know whether he's double muscled. I can probably tell just by looking at him that he's homozygous recessive for that. But if he was, however, a carrier, then I might need to do a test in order to determine whether he was carrying one of the mutant forms of that particular allele. And so we want to use these DNA markers in addition to other information we have on the animals, to help select the best animals from a genetic perspective, the ones that are most likely to pass on good genetics. And so you can kind of think of these SNP markers as little flags that are kind of spread throughout the genome that are associated hopefully quite close to genes that have an effect on the trait of interest. And so by selecting for the flags, you're pulling along that genetic variation in that gene so that you can select better which animals have good genetic variation for the trait of interest. So hopefully that gives you a little bit of background. So if, for example, this particular bull, I genotype him and I see that, you know, he's got no variation here, but at this particular location there's a G in the sequence here and an A in the sequence here, a T. So he's a GT heterozygote, and maybe, I don't know, T is associated with better marbling and G is associated with less good marbling. Then half of his progeny will inherit that T and half will inherit the G. And of course, if he's crossed with another animal that's heterozygote at that loci, and let's just say that the G was good for marbling, then a quarter of those animals would get both good forms of the marbling gene, good. Half would get the heterozygote form and a quarter would get the TT, the two bad forms of the gene, or get unlucky in the genetic roulette. And it's really this, what's called Mendelian segregation, that genetic testing can help us with, because it tells us where we have two parents that have a bunch of flush mates. We don't know whether they happen to get, you know, the two good genes or the two bad genes or one of each. And so by doing genetic testing, we can say, oh, look, that one got two good forms. This is going to be the better flush mate of all of these animals that otherwise we have the same parents, and so we don't know how different they're going to be genetically. And so it looks into this Mendelian segregation by doing genetic testing. So the other thing that's really important to understand is the difference between characteristics. So there are some characteristics that are totally driven by genetics and that are very simple traits. And perhaps the simplest one is gender. If you get a Y chromosome, you're going to be male. If you get an X chromosome from your dad, you're going to be female. It's 100% determined by genetics. There's no environmental interaction there. Other traits of this nature are things like coat color, certain genetic diseases like curly calf, double muscling, the presence or absence of horns. If you're heterozygote for horns, you don't get this type of a phenotype, right? You're either totally polled or you grow horns if you get the double recessive form. So those types of traits are really easy to work with, and they're really satisfying to work with as a geneticist, because I know exactly what I'm going to get. The more difficult traits to work with are the ones that are more economically important, of course. And they are these quantitative traits, like marbling, for example. And the reason is that there's a genetic environmental interplay there. And so you might have the best genetics in the world, and you're located out here in the desert in New Mexico. You may not marble terrifically because your environment isn't giving you the nutrients you need in order to get there. And so these genetic and environment traits typically are controlled by many, many genes, these quantitative traits, and you have this genetic environment interaction. So originally, when we first started out with tests for quantitative traits, and I think Dr. Gibb and I have been around since the first traits got introduced, we only had maybe 1 or 10 SNPs associated with those traits of interest. And they were coming out in early 2000, the early noughties in American, in Australian terms. Tests for things like meat tenderness. There was a couple of markers in kelpane and kelpostatin, and some for marbling that you may remember, GeneStar out at the time, feed efficiency, milk yield, disease resistance. And if you only have 1 or 2 markers for these types of traits, you're only really tracking 1 or 2 genes in these multigenic things. And there was kind of a disconnect between the DNA test, which maybe, let's just say, it was explaining 15% of the genetic variation in a trait, which is actually a fair bit for a single SNP to explain. That would be an amazing SNP. But if you think about the fact that in marbling, we've only got 37% heritability, in other words, genetics only contributes 37% of the variation. If you've only got 15% of the variation being explained by the DNA test, then you've only got 15% of 37% being explained by the DNA test, and you have this huge environmental component, and so your test results weren't necessarily matching your phenotypic observations, and it kind of made people a little bit suspicious of it. And the problem was that we weren't accounting for sufficient variation with those tests in order for it to have an obvious relationship between the phenotype and the DNA test results. And when you're only explaining that amount of genetic variation, you can't make a lot of progress because you're not really accounting for very much. And so what we've found is that you need to have lots of markers, and when I say lots, I'm talking tens of thousands of markers. And really the development of the SNP chips in 2009 and since then, originally the 50K or 50,000 SNP chip, which basically tracks 50,000 separate SNPs throughout the genome. So you can think of it as 50,000 SNPs spread through the 3 billion base pairs that make up the genome. That's really where the technology took off, and since then we've now got an 800,000 SNP chip that's getting used for research purposes, and in fact there's a whole bunch of groups working on doing whole genome sequencing of high-profile AI bulls, so getting all 3 billion base pairs sequenced. So this technology is moving way faster almost than the understanding of how we might best use it in a commercial setting. And so when this information first started coming to the producers, there was a group called the Beef Improvement Federation that works on genetic evaluations for beef cattle in the United States, and they were wanting to make sure that the DNA tests came into national cattle evaluations in a way that it helped to inform the genetic estimates that were being developed for beef cattle by the Breed Association. So they're the expected progeny differences, or the EPDs that you see in bull catalogues. And so there was a real effort to put together a group to say this is how this technology is to be implemented. It shouldn't compete against EPDs, it should be brought into EPDs, and that has happened. And so now what we have are these genomically enhanced EPDs that the breed associations are producing, and I'll explain a little bit, and I think Jim will too. But basically now if you have a young animal, you'll know his pedigree, so who his parents were, which will give you some idea of how he's going to perform, just based on likelihood of receiving good genes from mum and dad. You might have some performance records on him, so maybe some ultrasound information, birth weight, weaning weight. If he's an older animal, you might get some progeny tests, and that'll really increase the accuracy of this this merit estimate. But here we've got the genomic test result coming in as just another piece of information that's associated with the EPD, and the importance of it is associated with this accuracy, or R, which is basically the correlation between the genomic test result and the actual genetic merit. And if R was 1, it would be a perfectly predictive test of the genetic merit. We're not there yet, but we do have some R values that are getting up beyond 50%. So we are getting to where we can explain much more than 15% of the genetic variation, more like 50% of the genetic variation with some tests. And so that's kind of how it's being implemented in industry. And so, for example, the American Angus Association every week runs genetic tests with their genetic evaluation and spits out new, more accurate records on young sires that have had their DNA testing. And the correlations that they've been working with using the 50K prediction are in the range of 0.6 to 0.7, which if you've got 0.7, you basically square it. It's explaining about 50% of the genetic variation. It's a little lower for traits like milk and heifer pregnancy, and they're basically getting incorporated into the 15 EPDs that the American Angus Association releases. One thing we weren't expecting, I think the community, when we first started this, was that the way that the Angus Association developed those tests is they had large training populations with genotypes and phenotypes in their breed database. And they had phenotypes, or recordings, on traits that breeders record, so weaning weight, birth weight, that type of thing. And they had thousands of records, and they could basically use that to tell which DNA variation was associated with different traits. So you need these large training records in order to what's called develop the prediction equations. And that's what enables them to get good predictive ability on an unknown DNA sample going into the Angus Association. You get these fairly high correlations for these different traits. What we weren't expecting is that if you train in a black Angus population, so you use black Angus phenotypes and black Angus genotypes, that it wouldn't work very well when you went to another breed's predictions. So if you use that then in Red Angus, the accuracies drop off precipitously. And we weren't really expecting that that was going to be the case, and it's become evident that that is true. And so if we look at the kind of the genetic relationship of the different beef breeds, here's Red Angus and Black Angus. They're about as close as you can get on the Bos Taurus line as compared to some of these others, Gelbvieh and Simmental, and here's Hereford's way over here and Brahman's way out here. And so what that more or less told us was that we are going to need to develop these large training populations in each breed that were interested in genotyping. And so I think we went through this kind of hype cycle where we thought we'd be able to train once and use it on all animals, and that obviously wasn't true, and then we kind of slid down to the trough of disillusionment. But slowly over the last few years, the breed associations have been putting together their populations in order to enable prediction equations for their particular breed. And so at the current time, we now have a number of different breeds that are put together. Red Angus has 6,000 odd, Angus has 3,500 odd, Hereford 3,000 odd. And basically, we're getting up to more than 1,000 records in the training population. And you can see that the accuracies are getting up to that 0.5, 0.6 range. They're lower with less animals in the training population, so the more animals the better. But we now have it incorporated in various, the other breeds are now incorporating genomic predictions in the US beef industry. So I've mentioned Angus. There's also Charolais, Hereford, Simmental, Gelbvieh, Limousin, Red Angus are all doing it, and others are just getting their training populations put together. And so we're in a pretty good situation now as compared to where we used to be, because we can basically do genomically enhanced EPDs in almost all of the major beef breeds in the United States.